Do you know what is volumetric analysis? Titration is an analytical technique that is widely used in the food industry. It allows food manufacturers to determine the quantity of a reactant in a sample. For example, it can be used to discover the amount of salt or sugar in a product or the concentration of vitamin C or E, which has an effect on product color. There are three types of titration, which are simple titration, back titration, and double titration. A simple titration is a procedure that uses a known solution to determine the concentration of an unknown solution. Usually, until the reaction is complete, titrant or known solution is added from a burette to a known quantity of analyte or unknown solution. It can be further grouped into four sub-methods, namely acid-base, redox, precipitation, and complexometrics. Second is back titration. The concentration of the analyte is calculated in this type of titration by reacting it with a known amount of added reagent. Additional reagents are then titrated using different reagents. The result of the second titration reveals how much additional reagent was used in the first titration, allowing the concentration of the original analyte to be determined. And lastly is double titration. It is similar to simple titration. The only difference is that it involves two titration with several components in the titration or a solution of unknown concentration. Since there are two endpoints during the titration, two indicators, usually phenolphthalein and methyl orange, are used in these procedures.
First, let's take a look at the advantage. It is simple to execute and also very cheap. It also don't have any particular process to learn. It is simple to do. Therefore, you doesn't need to be skillful at this point. It's occur very swiftly. Moreover, it has highly accurate as well. It is much better compared to other titration method. Done with the advantage, let's have a look at the disadvantage. As we know, this process is conducted in open system. It can be affected by certain factors such as pH, humidity, and temperature. It may be most accurate, however, it is not always be accurate since there are chance of human error. If you got no reagent, the reaction will not occur. After getting the result, there is a lot of chemical waste produced that need to be disposed. The reaction only takes place in liquid phase. Let's move on to the next part, which is application of volumetric analysis in real life. Volumetric analysis is an important analytical technique used extensively in the food industry. It involves the measurement of volume of a reacted solution to determine the concentration of analyte in a sample. Some key applications of volumetric analysis in food analysis include adulteration detection, nutritional analysis, and sugar testing. Adulteration detection. The presence of adulterant in food like oil, milk, and honey can be detected through titration with specific reagents that react with the adulterant. This helps check for dilution, contamination, and other forms of adulteration. Nutritional analysis. Level of vitamin like vitamin C and mineral like calcium can be determined by titrating food sample with agent like iodine and EDTA. This provides reliable data on nutritional composition for labeling purpose. And the last one is sugar testing. The quantity of reducing sugar in food can be analyzed by titrating sample with Fehling solution. The volume of Fehling solution declares indicates sugar content. This method is used to verify and standardize sugar level in food. Thus, volumetric analysis provide the food industry with an accurate, convenient, and cost-effective means for quality assurance, safety, and nutrient monitoring of wide variety of processed food and beverage. The technique plays a crucial role in regulatory compliance and quality control in this sector.
In an acid-base titration, the sample is titrated with a standard solution of a strong acid or base. For testing acidity, the sample is titrated with a strong base like sodium hydroxide. The hydroxide ion react with and neutralize the acidic component like citric acid in the food. The endpoint is detected using an indicator like phenolphthalein. The volume of base required to reach the end of point allow calculation of total acidity present in the sample. The more sodium hydroxide used, the more acidic the food. For testing alkalinity, the sample is titrated instead with a standard acid solution like hydrochloric acid. The volume of acid needed to neutralize the alkaline component indicate the total alkalinity. This approach can detect excessive addition of alkaline agent like baking soda in food. It can also test for alkalinity in ingredients like eggs, dairy, and soy which influence their functional role in food formulation. Thus, simple acid-base titration allow reliable quantification of acidity and alkalinity level in all kinds of food and beverage. The method requires minimum sample preparation and provide rapid result, making it ideal for routine quality control and process monitoring in the food industry. Monitoring sugar and salt level is vital in the food industry to meet labeling regulation, quality standard, and consumer preference. Volumetric analysis offers straightforward, inexpensive method to quantify sugar and salt content. For sugar analysis, samples are titrated with a solution of failing reagent, which contain copper sulfate. Sugar like glucose and fructose reduce the blue failing solution to a reddish orange precipitate of copper oxide. The amount of failing solution decolorized correspond to the sugar concentration. Using the volume and noise concentration of failing solution, the quality of sugar in food and beverage can be accurately calculated. This method reliably verifies standardized sugar level in product. To determine salt or sodium content, the food sample first homogenized in water and filtered. The chloride ion in the extract are then titrated with standardized silver nitrate solution. Silver chloride precipitated as the chloride ion get complex by the silver ion. The endpoint is detected by formation of brown silver chromate precipitated when all chloride are consumed. The sodium content is divided from the volume of silver nitrate used, providing reliable data on salt level. Thus, relatively simple and inexpensive volumetric testing allow food manufacturers to precisely monitor two major food components, sugar and salt, compliant with nutritional label, health standard and quality control protocol is facilitated.
Determine water content or moisture level. Monitoring the moisture content of foods is critical to ensuring quality and safety. Volumetric titration provides a direct way to quantify water content or moisture levels. The basic principle involves removing water from the food sample, evaporation, and then titration the residue water. The food sample is precisely weighed, often dried, to evaporate moisture and reweighed. The wet loss gives total water content. For more accurate results, Carl Fisher titration can be used. The dry food sample is added to an anhydrous methanol sulfur dioxide iodine mixture. The water reacts with the Carl Fisher's reagent, consuming iodine. The iodine is the back titrate with a standard thiosulfate solution. The volume of thiosulfate required corresponds to the water content originally present in the food. This volumetric approach provides and exact and reliable measures of moisture percentage. It accounts for all forms of moisture and works for all food types. Monitoring moisture content ensures appropriate drying and processing of grains, nuts, and other foods. It also checks for loss of crispness in snacks, controls water activity in jams and jellies, and verifies correct moisture levels in dry or cured meat. Volumetric moisture analysis thus allows the food industry to standardize procedures, optimize quality, and provide consistent products with the right water content. It is an indispensable tool for regulator labeling, shelf life prediction, and process efficiency.